Okay, Karsten is supposed to be.
Yes, we are live.
Okay, perfect. And then, okay, so hello everyone, and thank you for joining us for another fine seminar. My name is Adriana Maldonado Chaparro, and I'm a professor at the Universidad de Rosario in Colombia. And I'm the host of the seminar this week. I'm really happy that you are all here today. And before we announce the speaker for this week, I would like to make some announcements. So first, I want to thank our previous speaker, Dr. Aura Raulo, for her awesome talk and discussion on transmission ecology of gut microbiota in the social networks of wild mice. I also want to announce our next speaker for next week, Dr. Sumana Anagiri. She'll be talking about e-sociality in ants. So we're looking forward to her talk. Just a couple reminders about the structure of FIND for those who are new in our seminar. There will be a 50-minute seminar, 45 to 50, followed by an hour of discussion. During the seminar, please make sure that your microphones are muted, and the only time that you should have them open is when we engage in the discussion with the speaker. During the discussion, please type in the question mark in the chat and I will call you on. And if you're watching the stream on YouTube, Karsten will help us relaying the question to the moderator or to speaker in this case. And as usual, we encourage the students to ask questions. So don't be shy and just please ask your questions. And then, with no further ado, I'm pleased to introduce this week's speaker, Chloe Fouilloux. She is a last year PhD student. She'll be graduating in June, so yay, from the University of Jyväskylä in Finland. Chloe's PhD research focuses mostly on studying the ecology and evolution of poison frog tadpoles. And more specifically, she was working on cannibalism in the genus Dendrobates. She's currently changing a little bit of topics, and she'll be working on disease spread and mechanisms of transmission in poison frogs as well. And so we'll look forward for her new work as well. Her publication record, it's really nice at her career stage. He already published 7 papers in different journals, such as Behavioral Ecology, American Naturalist, Ecology and Evolution, and Journal of Experimental Biology, among others. She's also been very successful at securing funding for research and has been awarded several grants from Oikos, the Company Biologist, European Society for Evolutionary Biology. She's also a Fulbright Fellow and some other grants that she's got. So if you want to learn more about Chloe's work, please do not hesitate in contacting her. Her seminar title is A Tadpole's Guide to the Galaxy, Communication, Kin Discrimination, and Cannibalism in Poison Frog Larvae. And so, Chloe, thank you very much for accepting the invitation, and the mic is all yours.
All right, wonderful. I will start with sharing my screen, which is the typical protocol. Do that, then share, and then get us up to present. Yes. Okay. How are things looking? Good?
Good.
Brilliant. So if you guys came here for Viviana Rojas today, I am here in her stead. I will be presenting some of my work today, some of our work also. And so today I'm going to be giving you a bit of a dip into the poison frog world, and more specifically into the tadpole world. But first, let's start things off with just considering how parents make decisions about where to breed, because harsh environmental conditions It can range from just being food limited to actually being natural disturbances such as fires and floods, which really challenges the survival and breeding success of animals. And so consequently, these challenges really trigger adaptations in the life histories of these animals that are affected by this. So we can think about how an animal makes a decision to breed, and actually that all of these decisions are subject to environmental constraints. So for example, in burying beetles, a harsh nutritional environment is going to affect actually the cooperation between parents. They can go from being female parental or male parental or sometimes even biparental care. It can affect the filial cannibalism of the brood. It can affect the duration and intensity of the care. And then in birds, we see examples of how, their nest choices affects their exposure to predators. And of course, the quality of construction of such is really can affect clutch survival. And so when we think about parental care in animals that are providing care and thinking about where to place their young, our thoughts are often constrained to terrestrial animals. But aquatic animals and amphibians in particular are no exception to this. When we think about breeding in amphibians, we probably often think about temperate species that breed in permanent bodies of water, like streams and lakes. And those are really great because there's a ton of resources for tadpoles, but also lakes have fish, and fish love to eat eggs and young tadpoles. And so the predation risk in these systems is extremely high. From an evolutionary standpoint, we can think about that, explosive breeding has arisen in order to kind of inundate and combat these high predation rates. But there's certain clades of frogs that have opted for a different solution. They've gone to become terrestrial breeders. And this is quite fascinating because, as most of you have to know, tadpoles have gills. So to breed terrestrially comes with a lot of interesting physiological constraints as well. So these terrestrial breeders use isolated points of water in the vegetation, and we call these phytotelmata. And that's just a really fancy word to say vegetation that contains water in a non-aquatic habitat. And so that can be fallen palm bracts, that can be tree holes, that can be the axil of a bromeliad. They range enormously in composition, in size, and in community. And that's actually what's the most exciting part about this, for me at least, and how a lot of my thesis work is built around all of these things, because we essentially have, maybe I should actually hide this. Can I hide this? Hide. Yeah, perfect. We essentially have these natural mesocosms that just exist throughout space, right? So we can go and see how the community structure changes, how the chemical properties of these pools change, where they occur in different habitats across space and time. And we can ask a lot of different questions about the animals that are there and how they change depending on where they occur. And while these little phytotelmata are wonderful at excluding large aquatic predators such as fish, they are no means paradise at all, because they come with a lot of environmental constraints. On what we can think about is, of course, we're escaping these large predators, which facilitates the evolution of parental care. But these small pools of water are really a balance between excluding large predators, but also being tremendously food limited. It's not like the community or the world within a palm bract is exactly teeming with life. So as a response to that, a lot of animals that have evolved to use these small pools of water have evolved a lot of cool adaptations, either in their adult stages or in their larval stages, to survive this. And so for our terrestrial breeders, parental care is something that we see a lot and it can sometimes be extended from the oviposition stage to up to the tadpole stage. And then we can also see it just occur at certain bits throughout development. One such example that we see is this clutch guarding by glass frogs, which I'm going to show a brief GIF of here. And here we can see fathers actually spend an incredible amount of time next to recently laid clutches, and they swat away incoming predators like wasps to protect the well-being of their offspring. So when we think about parental care, we might not really be thinking of amphibians, and that's fair. Most of the parental care we see, in mammals, for example, is fully female, is fully expressed in all mammals. And we see it a lot in birds, but actually 10% of amphibians demonstrate some form of parental care. And like I mentioned before, it can be quite short-term or even longer. But today we're going to be focusing on a certain clade of amphibians called, surprisingly, the poison frogs. And so I'm going to be just introducing them a little bit briefly, just to give you guys kind of a background on what they do and the diversity that they have, because probably when you've first thought about poison frogs, the first things we think about are their toxins and their colors. My first interaction with poison frogs was in Guyana when I was in the university, and I watched them and being territorial in their bromeliads, and from there I couldn't really stop working with them. They're absolutely fascinating. So when we tend to think about poison frogs, we tend to think about their adult forms, their bright coloration, the aposematic signal, how that works as a function of sexual signaling or as a anti-predator mechanism. And those are all well and good. That's great. But poison frogs aren't just adults. They go through a whole larval stage that, needs to be appreciated a little bit at least. And so that's what I'm here to talk about today. I'm here to talk about the larval forms of these poison frogs, because they have quite interesting lives as well and have evolved quite many adaptations and ways to navigate their environment that's worth thinking about. And so specifically, I want to first just mention the kinds of parental care that we see in these poison frogs that have parental care. So firstly, we have the transport of tadpoles from adults. So as I mentioned, these frogs are terrestrial breeders, which means that when these tadpoles hatch on the ground, we have an issue because they've got gills. And so what has happened is that these adults will carry their tadpoles. Some species carry them singly, some species carry the entire clutch, and they carry them on their backs. And they transport them to small pools of water, those phytotelmata that I was talking about, that serve as their nurseries throughout the rest of development. And what's really cool is that we have some poison frog species where the females do all the care, some where just the males do the care, and some that share biparental care. So there's a lot of cool comparative studies that are coming out in the poison frog care world. And on female care, it's because I mentioned these pools are food limited. Some species have actually evolved a quite fascinating way to overcome this food limitation, which is to supplement their larval diet with unfertilized nutritive eggs. So females of these species will come to different phytotelmata dispersed throughout the jungle and regularly deposit unfertilized eggs in pools to supplement, or sometimes their larvae completely depend on these eggs to complete development. So this has a lot of implications for the spatial memory of frogs, the navigation of frogs, and also the benefits that frogs and tadpoles acquire from acquiring these nutritive eggs throughout development. But the species we will be talking about for the rest of today is Dendrobates tinctorius. This is a poison frog that is found along the eastern Guyanese shield. I specifically work in French Guiana. They're not the most poisonous of the poison frogs, but if you lick them, you'll feel a little tingly. They've got an incredible diversity of back patterns, which allows us to identify them on an individual level, which is quite important for marker capture studies and being able to think about the evolution of color and pattern diversity. In fact, this species adult form has a ton of different forms that occur throughout Guyana. And all of these are indeed one species because they can hybridize, but the species that the population that we work with specifically is this Nurag population, and they're kind of found here in the center of the country. And they're thought to be in a hybrid zone between multiple morphs. So that's going to have a lot of exciting implications in the future. But today we are just going to be focusing mostly on the poison frog, Dendrobates tinctorius tadpoles. So just to do a quick wrap up of what these dads have to go through, because we have male parental care in this system, is that we have, again, these terrestrial egg clutches, these 3 to 5 eggs that are deposited in the leaf litter. And then after a period of 2 weeks or so, the tadpoles hatch in the leaf litter, which again, we are starting to have a problem because pretty much as soon as they hatch into the leaf litter, they are suffocating. And so as a response to that, we have fathers that are transporting their tadpoles one at a time on their backs to their nurseries. And so what we can see here is, for example, a dad, and in the foreground here, you have the eggs, and then you have the dad transporting his tadpoles. Usually this happens singly about 80% of the time, but sometimes lazier dads can carry 2, very rarely 3. But the sole purpose of this care is to transport their tadpoles to their pools of water, where they're then confined throughout the rest of development. So I wanted to show a quick video of what that looks like. You'll see this is a tree hole here. You've got a dad with a little tadpole hanging on his back, and he's hopping around, and he's going to go and deposit this tadpole. You'll see him kind of inspecting the surroundings. What's really interesting is that sometimes fathers will visit a pool and actually not deposit their tadpole, which leads us to think about how they're assessing that space, and that they don't always commit to it. But it's also important to think about that frogs don't just exist on one axis. These guys especially work on the vertical axis. So dads will sometimes actually transport their tadpoles up to the tops of canopies. And so not only are we seeing them invade phytotelmata on the horizontal axis, but sometimes they'll climb to more than 20 meters high into the forest canopy to deposit their tadpoles all the way to the tops of trees and small pools of water that are quite an effort to reach. And yeah, this is a great way to just kind of show you guys how far up they actually climb. And it's pretty impressive because they have to shuttle, like I said, 3 to 5 tadpoles once they've hatched, and they carry them one at a time. And I just want to take a moment just to highlight the energetic expense of that. So if we have one poison frog tadpole, going 20 meters high, I'm 1.64 meters. That's equivalent to me climbing, all the way higher than the Shanghai Tower. And you have to think about, of course, the force of gravity is maybe not equivalent, but it's worth saying that for these guys, this trek up to the top of the canopies is really an energetically intensive parental care behavior. And so that's, of course, a direct cost of care. But there's actually a lot of indirect costs of care that males face when they're caring for their young. And so, for example, when they're caring for their young, they're losing potential mating opportunities because they're on their one-track mind of carrying their tadpole to where they need to go. Also, what we see is that males will have multiple clutches in nature where they have to care for them at the same time. So the more time he's investing into one tadpole, the rest of the clutches and even tadpoles within a clutch are facing a higher risk of infection and of predation, and also, of course, of just drying out, because these tadpoles, once they're hatched, they need to get to water within a certain amount of time. If not, they'll just dry out and die. So really, these fathers have to be quite thoughtful maybe is not the word, but they have to be quite expeditious in their care, because the costs of care can be quite high. Like I mentioned, fathers are able to occupy a bunch of different kinds of phytotelmata. Some, of course, from a clutch can go to more than 20 meters of vertical height. Sometimes he can just, go to a fallen tree. Sometimes he just goes to palm bracts. But all of this to say is that these dads have these clutches dispersed throughout the forest. And so he has to kind of have this mental map of where his eggs are, has to have the countdown calendar of when they're hatching, has to be able to revisit this clutch that has recently hatched to transport all of those tadpoles. And so he has to have quite an interesting and complex cognitive ability to be able to orient himself and also transport his tadpoles throughout the forest. When we looked at this by following the frogs themselves, Our collaborators, Andreas Pachucones, who led this study, looked at the territoriality of frogs and where they go when they transport their tadpoles. And actually, dads not only go farther than their territorial boundaries, but they actually don't even use the closest pool available to them. So they'll go farther than expected to bring their tadpoles to their deposition sites. So they, again, are putting in this incredible amount of work for parental care to get to these sites, which we are now we'll talk about a little bit more, but they're very non-random. They select certain sites, and that's quite fascinating. And I just want to make one point again about the spatial capability of these frogs. In a recent publication, what our collaborators led by Andres Pachucones did is that we did some translocation experiments. So from home, translocating, taking the frog outside of their home range and seeing if they have this homing behavior. And so what we can see of tinctorius for the adults on a 50-meter scale, the homing behavior is quite high for both males and females. And so then they repeated this on the 200-meter scale. And here we see a little more variation in the ability of these frogs to orient themselves home. But all of this to say that the mental map of these frogs in navigating the jungle really warrants us asking, why are they going so far to get to these pools? And so what we did then is that we looked at a lot of pools across a couple of years, and we looked at phytotelmata from the ground to those that were, a couple meters high to those that were more than 20 meters high in the canopy, in order to kind of see what kind of spaces these tadpoles were occurring in. And so there's multiple pool properties that we looked at. This first, of course, was just the physical, how big was the pool? How high was it? How much water can it hold? From a chemical perspective, looking at the pH, the alkalinity, salinity. And then, of course, from a biological perspective, we are looking at the presence and absence of other predators, the kind of insect diversity that there occurs. And what was really fascinating across all of these data, across all of these years, is that tinctorius have a preference. So when I ran a principal component analysis, we see that they tend to occur in pools that are higher in vertical height, have a higher hardness and salinity and more insect diversity. But really what I want to highlight here is that there's huge flexibility in where these guys occur. And I'm going to kind of show this kind of within a community scale quickly to kind of anchor how important this is in comparison to the other species that we looked at. So when we look at the kinds of species and where they occur on the vertical gradient, a lot of species are very predictable in their pool use. This can be because they're physiologically limited. If we think about this little brown poison frog here, Allobates femoralis, they're not great climbers. So it makes sense that a lot of their tadpoles are confined to just, terrestrial pools. But there are other species that are arboreal, or at least semi-arboreal, and again have very predictable patterns of using pools of a certain size or that occur at a certain height. In comparison to that, we see that tinctorius are completely flexible when it comes to vertical height. They use pools that occur on the ground to more than 20 meters high. We see in terms of chemistry, it's quite amazing. These tadpoles can withstand a pH from about 3 to more than 7. So the kind of chemistry these tadpoles can tolerate is also quite amazing. And then abiotic factors, just in terms of temperature and the actual volume of pools. Again, these tadpoles seem completely unconstrained. And so we have to wonder, why do dads use such diverse pools? Why would he put his tadpoles in pools that can evaporate within a day? And so when we think about the entire transect of species that use these kinds of pools, right? The ones that are fully terrestrial, the ones that are phytotelm specialists, the ones that only stay up high, we have one species that can use all of them. And actually, that's pretty great, because dads can kind of start bet hedging and using different pools that might have different benefits depending on the conditions. For example, let's zoom in on the tadpoles that occur higher up in pools in the tree canopies, right? let's look into those ones. Well, we tend to see them occur with a species called Trachycephalus. Those are these guys up here, the Amazonian milk frogs. And the thing is that these guys lay their eggs in the pools. And so when they hatch out, it's an all-you-can-eat feast for all of these tinctorius tadpoles that have been carried all the way up to the treetops. So perhaps in that way, it's worth it for dads to carry their tadpoles up to these pools that are occupied by other species. We also know that these pools tend to be deeper. And when we think about the evaporation potential of an entire nursery disappearing in 12 hours, well, maybe it is worth the energy to climb all the way up if you have a pool that's 100 liters deep. Of course, when you exist in an ephemeral world, it's pretty important to get out of it as soon as you can because the risk of dying is high. So it again makes sense that if we start looking at the communities that are in these different pools, maybe dads are choosing these pools that have more mosquito larvae, for example. Or have the tadpoles of other species that can be great sources of protein for our tinctorius tadpoles to grow, reach metamorphosis, and escape these ephemeral conditions as quickly as possible. So, like I was saying, these higher phytotelmata tend to have larger volumes, which is tightly linked to stability. And parental care isn't energetically expensive. So why not play the odds of probability to increase success? If you carry one tadpole to the top of a tree, deposit one in a fallen log, and one in a palm bract that's right next door, dads are kind of able to maximize their investment in care and also the probability of at least one of their offspring reaching metamorphosis. And so from the parental perspective, that's kind of the story that we have to tell, or that I want to tell in this point, in the sense that we've got diverse choices for adults, right? These tinctorius adults are essentially unconstrained when it comes to where they can bring their tadpoles. Yet they tend to make risky decisions for offspring development. They deposit tadpoles in pools that might evaporate overnight, or during the day rather. And then also something I'll get into moving forward is that these tadpoles of this species are aggressive cannibals. So not only will he deposit them in shallow pools, but he'll also deposit them in pools occupied by large cannibals. And so as a result of these decisions, really the rest of the story are how did these offspring mediate the choices made by their parents? What do they do? Because now from the tadpole perspective, you are confined to a phytotelmata for the rest of your larval life. There is no escaping. There's nothing you can do. So like I mentioned, from a chemical perspective, they're there in situations that can be, from a neutral pH all the way to an acidic pH. They're in volumes of water that are incredibly diverse, and also they face this biological constraint of competition. And so I'm going to move now into into competition a little bit more, because these tadpoles are cannibals. And so the choice to put a tadpole in a pool that's occupied by larger tadpoles is maybe counterintuitive at first thought. If we look at what these guys look like in the wild, they're quite aggressive. This is a tadpole in a phytotelmata consuming another one. And so cannibalism is not only, it's not obligate, but it occurs quite frequently. And they can pretty much eat anything. The mouths of these tadpoles are meant for shredding and consuming and restraining. So they frequently eat the tadpoles of other conspecifics. They'll eat insects. They'll pretty much eat anything they can get their little beaks around. And so you might think that, okay, there's got to be perhaps that dads avoid putting tadpoles in pools where, the size difference is really large. Maybe they're going to make sure that, okay, if they deposit them in pools with other small tadpoles, at least they'll, the risk of cannibalism will be lower because they're all the same size. But that's not what we see at all. Actually, fathers seem to prefer, or at least the latency to deposition for tadpoles is much shorter when the size difference is greater. That's weird, right? And so then when we think about the probability of this size deposition, between the resident and an incoming tadpole, tads actually deposit tadpoles in pools that are occupied by residents that have much larger sizes. And so it seemed that this trend made us think about, okay, are dads actually feeding their tadpoles to other tadpoles? And if that's not happening, or if it is happening, what is the size structure within populations looking like? Because, you know, if we have pools that are occupied by 20 or so tadpoles, what's our size differences looking like here? What can we infer from all of these differences? And so we had a great bachelor's student named Joanne look at a bunch of pools that had a bunch of different tadpoles and measure the size of tadpoles within these pools. And what he found is that when there's fewer tadpoles, when there's fewer tadpoles within a pool, the size difference is much larger. So there's high variation, and which you would, you know, logically think that's an easy meal, right? If there's only 2 tadpoles and one's big and one's small, we can kind of see where this story is going to go. In comparison, in pools with higher numbers of tadpoles, we see that the tadpole size is much more homogeneous, but also much smaller. And so that makes us think, okay, they're more similar in size, but maybe cannibalism isn't happening here because they physically can't eat each other because they're of a similar size class. This really doesn't answer the question, though, of why do fathers deposit tadpoles in pools that are already occupied? And while I'll go into some other details later, the most obvious answer, at least from our perception, is that a large tadpole can actually be a cue that that pool has met the requirements for a successful development of a tadpole, that pool has had at least a consistent enough volume, consistent enough food source, that it might be worth it for a father to risk it and put a young tadpole in a pool, because that pool is stable enough to at least support the developmental duration of a different individual. And development in this species can last multiple months. It's not something that's quick, and so that can actually have a lot of value in terms of fitness when we think about the decisions he's making. But the other thing that we notice is that tadpoles aren't always cannibals. And so in this ephemeral world, in these pools where these guys develop, it's important to think about the evolutionary forces that are shaping these tadpole choices. Like I mentioned earlier, these pools are extremely ephemeral. So in the morning, they can be full, and by the afternoon, they can be completely empty. And so when we think about the way that these tadpoles acquire food, they don't get to have this specialized behavior in food acquisition, such as oophagy. They're not waiting for mom to come and deposit nutritive eggs. They kind of have to make their own food opportunities. And so when a tadpole is surrounded by other smaller tadpoles and chooses not to cannibalize, when they forfeit a decision to feed, that really becomes interesting for us, because that seems to indicate that these tadpoles are not just random machines eating anything that they encounter, that there must be some other forces at play driving tadpole decision making, especially when it comes to acquiring these super important nutritive sources. And so we built upon these field observations by thinking about how inclusive fitness theory can help us understand these cannibals' decisions. And so together with Lutz Frommerhag and Janne Valkonen, we were able to, in a laboratory setting, control the size, relatedness, diet of tadpoles, and then see how that's influencing their decision-making and their aggressive behaviors. So now I'm going to move in to really discussing what informs a tadpole's aggressive decision. So we, in this study, manipulated not only the relatedness between tadpoles, so we were able to control if they were full siblings, half siblings, or non-siblings, and also modulated the size between pairs. So some sizes within a pair, there was always a larger tadpole. And so really what we wanted to get to here was, does family matter to a cannibal? Does this also help justify these decisions that dads make that seem so counterintuitive in the field? Does it matter if they're paired with a sibling? Does it matter if do they even have the ability to recognize half siblings? And what happens when they're paired with non-siblings? in different species of cannibals, in larval salamanders, for example, we see that they have very fine scale kin recognition, and they even decrease aggression towards cousins. So we had the basis to thinking that perhaps that these tadpoles were able to recognize kin. And so what we did is that we put them together and we watched them to see what happened. This is just, I'm going to show you guys one panel at a time. And this is just the number of aggressive behaviors by tadpoles. We didn't have these trials run until cannibalism for ethical reasons. But in terms of biting and chasing, we see in non-siblings, this first panel, that the large tadpoles within pairs are the most aggressive, and the small tadpoles are freezing a lot and really not showing that many aggressive behaviors. In comparison to half siblings, we see a slight reduction in aggression, nothing that was significant. And actually, really when things get quite notable is then in the siblings, where we see the large tadpoles significantly reduce the amount of aggressive behaviors towards their small counterparts when they're full siblings. So what we're seeing here is an interaction between size and a relatedness. So when we approach this from an inclusive theory standpoint, we find that aggression is size dependent, but that it's also costlier for small tadpoles. So it makes sense that these small tadpoles are not going to be putting themselves out there because they're just putting themselves at a risk of being attacked and eaten. And so in that way, we see that tadpoles are not randomly swimming around and just attacking anything they come into encounter, that they actually are able to recognize their siblings and modulate their aggression in response to that. So in brief summary here for this part of our talk, we see that the large tadpoles are more aggressive than small tadpoles overall, but that these large tadpoles significantly decrease their aggression when their pair is fully related. So we see that aggression between tadpoles demonstrates that this activity is non-random. And that's really interesting when we start putting into context the kind of community that lives within these pools. So maybe instead, if this pool is occupied by other species of tadpoles, by other heterospecific insects, tadpoles might be able to live in high densities if they have these alternative food sources, because they seem to be able to recognize a kin. So when we think about the ephemeral world where they live, we have to think about how they're then managing predators in a dynamic context, because not only are tadpoles having to mediate relationships between conspecifics, but there's also other predators that exist in this ephemeral world. So for example, we've got our tinctorius tadpole, we've got large tadpoles, but we also have these small larvae that are adonata larvae. And so tadpoles will eat those guys. But in comparison, if we see these dragonfly larvae that are a different type of adonata, if they're big enough, they'll eat the tinctorius tadpoles. Of course, if there's other species of tadpoles in these pools, we've got potential food source for a large tinctorius, and those can also be eaten by these dragonfly larvae. And then, of course, we've got these smaller tadpoles, like we've mentioned before, that have cannibalism. So all of a sudden, we've got a multi-trophic network here, and these tiny little palmbract, right? We've got this ephemeral world full of complex dynamics, and tadpoles have to mediate all of them. And so when I really started looking into this and looking into pools and thinking about how they're mediating all of these relationships, it became quite clear that the environment itself should probably play a factor. And what I mean by this is actually the visibility of the pools. The clarity of the water can range enormously between phytotelmata. Some can be as clear as drinking water, while others can be very turbid, very deep red color. And so that got me thinking that has to have some kind of role in how a tadpole not only is developing in the darkness versus a clear pool, but also their relationships with con and heterospecifics. So we put forth a couple of hypotheses in a perspective piece, thinking that maybe tadpoles that are developing in these turbid pools are going to have less efficient foraging, And also maybe they're not going to be as good at avoiding predators. And then from a proximate mechanism, there might be developmental aspects that are affected by developing in these turbid, dark habitats. How is this affecting the development of eyes, for example? So we have the thought that's been substantiated through at least some fish studies that maybe the wavelengths and the spectral tuning of the retina would be red shifted in tadpoles that develop in these turbid environments, and so that they're better able to navigate these spaces. And so this was all great because I put forth a bunch of questions, but then we had to start with the first question of, do tadpoles actually see each other? Do they actually use vision to navigate their environment? Because sure, these pools might vary enormously in tint and turbidity, but does it even matter? Do they even use their eyes? And so in the lab, we had a brilliant master student pick up this question and kind of look at tadpole communication. How do tadpoles even recognize each other? How does this affect their activity? And so here what we have is one large arena full of water, and then within it we have 4 containers that contain our 4 different treatments. So here, of course, I'm only showing the control, and in this case we have a tadpole just swimming around in their own little container of water. They don't see anything, they don't smell anything, they're just living their lives. In a container that's in front of that, we have a tadpole that's been introduced to the olfactory cues or the chemical cues of another tadpole. So we essentially let another tadpole swim around in some water for a couple of hours, and then we place that water into this experimental unit, this arena within the arena for those chemical cues. And then we have a tadpole that's only using visual cues. So in this sense, they're able to directly look at the tadpole that's in front of them. As you can see, they have there's no opaque wall here blocking them. They're able to make visual contact with each other. And of course, that leads into us having our tadpole with bimodal cues. So they're able to not only see the other tadpole, but they also received the same olfactory cues of a tadpole that had been swimming around in the water previously. So what do we see? Well, we see that tadpoles from the vision treatment are swimming significantly more than the other experimental conditions. These tadpoles were swimming all over the place. And when we were able to actually look at the distribution and space use of these tadpoles within their respective pools, we see that these tadpoles from the vision treatment are spending a significantly higher amount of time close to the shared wall here. And so in comparison to the control tadpoles that were just kind of, chilling out in just their opaque corner. So these spatial patterns, when we look at them in terms of their X and Y coordinates and analyze the way that they use space, are significantly different between the control and the vision treatment. Our master's student right now, we're working on getting the data for these final 2 treatments, but I can tell you it's pretty exciting in the sense that it really seems that these tadpoles are not only moving around more when they have visual cues, but they also seem, based on their space use, to be looking at the wall towards the other tadpole. And so within that context, it seemed pretty clear that tadpoles are at least using visual information to some extent to navigate their world or to gain information about their world. So then when we put this together with the environmental aspect, we can now think about, okay, we know that they're using vision. We know that these worlds that they live in can range wildly in their turbidity. How does this affect their ability to see heterospecific predators and respond to them? And so in this sense, I was actually able to go again into the field and measure the turbidity of these tadpole phytotelmata, the phytotelmata occupied by tadpoles. Again, phytotelmata are amazing mesocosms for being able to measure these things. And so I was able to take a water sample, and I'll get into the quantification of that quickly after this. And I was also able to take a tadpole from that pool and use them immediately and see how they're responding to visual cues. I quickly want to make an aside of how I quantified pool turbidity, because it's quite, I think quantifying turbidity of a small microhabitat hasn't been frequently done. And actually in this study, I did a comparative study between 2 species. I'm only going to be talking about tinctorius today, but the reason why I bring them up just briefly is because for this Pomelio species, which is another phytotelma breeder, I was able to use a spectrophotometer to quantify the pool turbidity. So essentially we get all of the wavelengths, and the light that's refracted or absorbed by the sample gives us an idea of, the color of the pool. And I also photographed the same pools, and I was able to validate that spectrophotometry and our photographic method of quantifying pool turbidity yielded the same results. And so that's really a cool way within now the fieldwork. We don't have to use a spectrophotometer, which might be, really excluding some scientists from being able to do this kind of work. There's actually using ImageJ and pretty accessible photographic methods, we can reliably quantify pool turbidity. So within these assays, what I first did was put the tadpoles on a checkered background and just see, without any predator, what their preference was for black or white. And then after that, a tadpole was either put in a black or a white arena. And within the center of this arena, they were either faced with a large dragonfly larva, which is again a tadpole predator, or they were faced with a conspecific. And so each tadpole went through both permutations of the predator and conspecific across both different luminic conditions. And so let's just jump into some results. What we see here are tadpoles that developed in a dark or turbid microhabitat. When it comes to their activity, don't have any clear differences in responses to either visual stimulus. And so this was really fascinating to see, because these guys have been placed right in front of, a predator that could eat them, and there seems to be no discrimination. And that's really quite a stark contrast when we look at tadpoles that developed in bright and crystalline microhabitats. All of a sudden, we're seeing their response to conspecifics to be significantly stronger than what they respond in terms of Adonata larvae. So this not only shows us, again, that they're able to visually discriminate conspecifics, which we kind of already showed in the lab, but also that their developmental conditions had a direct impact in the way that they're navigating these predator-prey relationships. When we look at space use, this continues to be true in the sense that the zone 1, which is again closest to where that stimuli was, they're spending a lot of time interacting with this visual stimulus when it contains a conspecific, versus when it contains a predator, they swim away and spend a lot of time separate from that. So this again is showing us that the places where these tadpoles grow up really affects the way not only that they move, but also the way they navigate space. And when you think about a tadpole that needs to, subjugate other conspecifics in order to hunt, this has a lot of potential impacts with how these tadpoles that develop in darker spaces are going to be able to reach metamorphosis. What does this have in terms of implications in terms of survival of tadpoles, and what does this even mean for the carryover effects of what happens to their eyes? So overall, we were able in this part to really quantify these bright crystalline pools versus the dark turbid pools. And we find, again, an increased activity and spatial interaction with conspecific visual stimuli. And we see this both in laboratory conditions and in wild tadpoles. And then again, we see a reduced predator response when tadpoles are raised in turbid environments. And I'd like to take a brief moment to just emphasize that if they had developed in a turbid environment, in the experimental arena, the water was clear. And this was just a limitation of the time and the amount of individuals that we had available to us. But the fact that tadpoles that were raised in clear pools and then were tested in clear pools had strong differences in their response to different visual stimuli makes us think that if they were raised in a turbid pool and then challenged in a clear pool, could this be evidence for that proximate restructuring of the eye? Could this be evidence that something has happened in the retina and that they've become red shifted, and so that when they're in these clear environments, they freak out or maybe can't see as well? And that needs to be looked at, but at least we're seeing a clear developmental effect across different treatments. But in the future, what I would love to do is to have, tadpoles reared in a turbid environment and then also tested in a turbid environment. I would like to end this talk with kind of thinking about what we can learn about poison frogs. We've got a lot of questions about ecology, the kind of habitat they use, developmental plasticity based on the habitat where they're raised. We can also think about sociality between tadpoles, the way that they're making decisions in their interactions, and also the predator-prey dynamics within larval communities. There's, of course, parental care and conflict, so the way that fathers are caring for their tadpoles in terms of how care changes across development, even within a clutch. How does a father's investment into the first tadpole he transports change from the fourth tadpole he transports? Is he always making these decisions to go up to the tops of forest canopies, or is he sometimes, bet hedging and making modulating the amount of care he's investing based on what number this tadpole has, is in the clutch, if they're the first to be transported or the last to be transported? And of course, then you have this amazing navigation, memory, and spatial assessment by adults in terms of choosing developmental habitats for tadpoles, and also spaces where they occupy, across the vertical gradient. With that, we have a lot of people to thank. Thanks to Bibi for giving me this opportunity. It was a great 12 hours. And just a lot of collaborators, and also thank you guys so much for sticking with me and having this conversation. I look forward to talking more about poison frog tadpoles, and everything about tropical frogs would be great.
Thank you very much, Chloe, for a great talk on a very different topic, or at least the study system and the one we're used to here in our fine seminar. So yes, thank you. Well, if anyone has a question, please type in a question mark on the chat and we'll call you on. And if you are in the YouTube, please type in your questions in the YouTube. Karsten will be happy to just relay to the speaker. Let's see our chat. So here we have our first question comes from Kaya. Just a brief reminder, please state your name and your affiliation. We're trying to build a community and we are happy to get to know each other. Kaya?
Hi, I'm Kaya Tambak. I'm a postdoc at Hunter College. That was a really fascinating talk. I have tons of notes. Thank you for that. I'm wondering whether, so I was just asking myself, why would a dad deposit a tadpole in a pool with a resident that's not kin? And I wonder whether you think there's potential for some brood parasitism-like dynamics going on, where, because they must have a pretty good spatial memory and some kind of clock, keeping track of the timing of their depositions. And so maybe if there are some sort of like parasitic dads watching over how other dads are, going about, and then timing it such that if they have a tadpole ready to go, they'll go and quickly deposit one in a pool where another dad recently deposited theirs, and then, I don't know, eat that other tadpole or whatever, such that the other dad might not know that their next tadpole is going in with a non-kin, potentially cannibalistic tadpole.
That's a really, okay, so thank you for the question. I think I had never thought of it that way. Because the thing is, the territories within this species specifically are quite loose. And so then they can also travel quite far distances to deposit their tadpoles. I think the idea that dads could be kind of like watching and parasitizing is really fascinating, especially when we think about tree falls. So when a tree falls, all of these frogs just pile on to these potential deposition sites. I think because they're limited resources, but the idea that if you get there first, you have a big advantage because your tadpole is going to be larger they grow quite fast once they're deposited. So when you think about all the tadpoles that are deposited within a pool after that, I think it really has to do because there's such limited resources. And because the chances of being cannibalized are worth it if the pool is going to survive at least a couple of months. But I think also the deposition strategies being copied by dads is something we've thought about and something I would love to test with like VIE tags. If we could put like elastomers within the tadpoles and be able to track where they go, that would be so cool, because that way we would be able to see the decisions dads are making and to see if other dads are making similar decisions. But it's something I would really be interested in, but haven't gotten around to. But yeah, it's a great question.
Thank you.
Thank you, Kaya. Our next question comes from Linda Lanning.
Thank you, Chloe, for a very lovely talk. It was very interesting. So my name is Dendrobates Makoya, and I'm a PhD student working on the costs and benefits of solitary living in the bush collaret in South Africa. So first, I have a comment. It was very nice to see that you were able to validate a lab method so that you can be able to use it to use it more efficiently in a field without having the constraints of all the lab work that is needed for that. And I think that's really cool, and that's something that's very important, especially when one is working in a field. And then my question to you is just that when they were looking at the spatial range of the frogs, do you know what type of tools they use in order to navigate their landscape, and whether it's visual or smell, or what did they use?
Thank you so much. So there's a couple hypotheses around, and some have been tested in very informal ways. What I can say for tinctorius, well, I can't speak to tinctorius directly, but there was this thought that perhaps they were magnetically oriented, so that they used like birds do, I guess, magnetic orientation to be able to know where the pools are in relation to themselves. And I can tell you that at least for if you put a tiny magnet on a frog's back, it doesn't seem to bother them or disrupt anything they do. So I have no idea. I can't speak to it in any kind of formal way, but at least I don't think it's magnetic. But perhaps smell. I know Andres Pachucones and Jennifer Serrano Rojas have recently published that another kind of poison frog, they're able to locate pools based on stagnant, kind of stinky leaf litter. So perhaps there can be some kind of olfactory cue going on there when it comes to being able to at least locate the pools. But then again, how they actually keep a mental map of the jungle, and again on 3D, because they're also climbing up the trees. No clue.
Okay, all right, thank you.
Thank you, Linda Lanning. Our next question comes from Birgit.
Hello, thank you so much for this really interesting talk. I'm Birgit Sabo. I'm a postdoc at Eva Ringler's lab in the University of Bern in Switzerland. And I wanted to know a little bit more about how you raised the tadpoles before you did the kin half-sibling discrimination task, because I'm planning to do similar things in Femeralis, and I just wanted to see what you did.
Of course. Yeah, so pretty much, we have our suite of breeding couples. We have, I think at the time we had 12 terraria with a couple in each terraria. And from there, just, building a breeding network to have certain amounts of siblings, half siblings, and non-siblings by doing, the switches in a consistent enough manner. And then you kind of start building up a giant tadpole colony. And then from there, just making sure that they're all fed exactly the same amount across, I was feeding them, I think 3 times a week or so. And then once they're paired, at least for us, we had to make sure to separate them before they cannibalized each other. So just having to set up kind of a visual a video system, because sometimes, you know, I left them for an hour and that's when I watched directly. But then if I watched for 3 hours, I'd have to, you know, I had a Zoom call with the tadpoles to see what they were doing. So yeah, pretty much it, as long as your frogs are happy and breeding, was one of the easier experiments that I did.
I'm more concerned about, so I'm assuming to track individuals, you had to separate them physically while raising them, or did you just use them immediately after they hatch? Or did you, for example, after they were transported to water, did you raise them for a certain amount of time before you can use them, because they're pretty small, and when they're so small, it's quite hard. They might die from the stress of doing these experiments. So usually you raise them a bit. So did you then raise them in visual and olfactory isolation from other tadpoles, or were they like in groups? That's what I'm interested in.
I see. Yeah, so raised them from clutch hood, so we pretty much took the clutches out, separated. Once the tadpoles hatched, then they all grew up in their own individual little containers. And then they were only tested for, you know, for that experiment that I lined out that our master's student did for that time. So in all other time during development, they were just growing up alone in a, you know, in an opaque pool, so that they didn't have, because, you know, if you grow up seeing other tadpoles, I think that's going to have a big effect later in life. So we kept that at least until they were. Yeah.
Thank you so much. Thank you.
Thanks.
Thank you, Birgit. Our next question comes from Luke.
Hey, I'm Luke Lauter. I'm at the University of Texas at Austin. I was curious with the kin recognition stuff. Do you think the dads are facilitating kin recognition amongst their subsequent offspring in any way? Like, are they giving any signal when they get there that could, you know, inform the tadpole that's already there that, hey, this is one of your siblings, don't eat them? Or do you think, is their eyesight sufficient that they could recognize their dad out of the water or something? Is anything like that going on, do you think?
That's a fascinating question. I would say from a gut instinct, no. A lot of the kin recognition that I did here, I had actually, like I mentioned to Bridget, hand raised all of those tadpoles. So they never met their fathers. They grew up in a fatherless home, and they were still able to recognize each other. At least, and then in that test, I wasn't controlling, I wasn't separating the cues. They really just, you know, were in the same pool of water and decided to attack each other or not. So I think really in that sense, the olfactory chemical aspect of shaping aggressive decisions is what's going on for tadpoles. But yeah.
Okay, that makes sense. Thanks.
Thank you, Luke. Our next question comes from Amir. I don't see him.
Hi.
I think he's...
I'm actually Mia's student. Was my PI. My name is Pramik. I'm a PhD student. I study social networks and rock hyraxes in Israel. So I have a very broad question. Is this species, are the tadpoles more precocial? Are they more highly developed than, you know, other sympatric species or other poison dart frogs that they live with? Just out of curiosity.
I wouldn't say so. I guess. Can you repeat the question?
So are they more precocial? So are they more, you know, are the tadpoles, when they're being put in these pools, are they more advanced in development than other sympatric tadpoles? Or
like speed of development?
Size,
yeah.
size, age.
Yeah, there's some of the bigger species definitely compared to other guys. They're definitely, especially if they've had a protein rich larval life, these guys can get pretty chunky, like a gram, which is maybe hard to visualize, but you know, if this is like a big chunky tadpole, there can be some that are, you know, the size of my, like, you know, tadpole, my pencil tip. So yeah, they really can end up being quite dominating in their larval life compared to other species. But actually at size of transport, they're not much bigger than other comparative species. I'd say compared to toads and stuff, they're bigger, but compared to other poison frogs, at least upon transport, they're within, the same size class. But then throughout their larval life, they can really get quite large. And the variation in size there is quite impressive as well.
Okay, thank you so much. Yeah, that was, yeah, thank you.
Thank you very much. And our next question comes from Tomislav.
Hello, I hope you see me. I'm not sure, but you hear me.
We see you
Yeah,
and we
good.
hear you.
Yes, I have actually two questions, or a question and a suggestion. The question is what really perplexed me was when you said that you were forced to stop your experiment because of ethical reasons, and I was kind of perplexed. I mean, do you have ethics for amphibian tadpoles?
Oh yeah, lots of.
So okay, so they can. So you must have written permission to allow amphibians eat each other. Is it so?
Actually, we weren't even allowed to let them eat each other. We were allowed to let them bite each other until. But then we had to stop the trials if there was any kind of permanent or lethal damage. So.
Okay. So thank you very much. That is a very very illuminating answer. The suggestion was that there is actually the centrality closeness analysis, which use a Bayesian approach, which could be quite fruitful, I think, because the centrality closeness with Bayesian approach is not dependent on the sample size, as frequentist approach is. So, I mean, it was just a suggestion. For example, the Smith and the Reinhardt have done it in investigation quite recently on a sperm competition, independence of size in Rhodeus amarus in the fish. So maybe it could be a fruitful approach, but.
So does it use, like, when you say centrality closest, is that within physical space or within?
Yes, of course. That is actually based on the closeness of the individuals consisting the population, and I have not tried it, but I've learned about it quite recently, and it was just a suggestion.
Yeah, I think
centrality closeness, which is quite independent of sample size, because as I have learned from your talk, it is quite small samples that you are dealing with.
Yeah.
So maybe it could help.
Yeah, absolutely. And also, when thinking about that, when we estimate territory sizes in frogs, especially frogs that can climb, it's super difficult because we can estimate territory on a horizontal distance, but how do you incorporate the 3D component of it? Because there's also the climbing, and that's a whole. So that's
and especially if each father is taking one kid at a time, it's quite small sample, isn't it? Yeah. But that was my suggestion. Thank you very much for an interesting talk.
Thanks.
Thank you, Tomislav. Our next question comes from Karsten.
Yes, hello, I'm Carsten Schradin from the CNRS in Strasbourg, France, studying mammalian social evolution. Yes, thank you, Chloe, for a very fascinating and enthusiastic talk. It's always amazing to see how these fields developed, poison frogs research, and people coming up with new, interesting questions and fascinating results like this here, topics not addressed before. I have one comment and one question first. Just because there are new things coming up, but I was also reminded of old results like mental maps as a very highly advanced cognitive skill that evolves independent of sociality, while these days most people are studying cognition related to group living, which is quite interesting for me. But my question is regarding to one of the remarks you did at the very beginning of your talk, saying that some dads, you call them lazy dads, carry 2, maybe sometimes even 3 tadpoles. So this implies that it's really an individual trait, that there's consistency, personality, that some dads always carry 2 and others always 1. Is that true, or was this just a general statement?
No, that's a great question. So I have to say, so I have no idea if this is a personality trait, and it's something that's really interesting, but I think what that assumes is that that's from the top down, and that the fathers are controlling the tadpoles getting onto their backs. And I would actually flip the question and say, are the instances where dads are carrying 2, sometimes 3 tadpoles, are the tadpoles wiggling on to the dad's back saying, please get me out of here? Because when we think about, there's parental care, but also there's parental conflict, because when we see them trying to deposit, sometimes dads will go and deposit tadpoles into pools. Sometimes they'll really be trying to shake the tadpole off, and a tadpole won't release from the dad's back. And so I think, it could be lazy dads in the sense of, I mean, I guess it really depends. And I don't know, to be completely honest, who's controlling this relationship. And I think it might be a little bit of both. And it's something that's always really fascinated me that I've never been able to really crack open, but it's really worth asking.
Okay, thank you.
Thanks.
Thank you, Carsten. Our next question comes from Suleima.
Yeah, hi. Absolutely wonderful talk. I really enjoyed it and was just glued to my iPad listening to your talk, but I did miss about a minute because I had to go deal with one of my dogs. So you may have mentioned the question I'm going to ask, but I guess I should introduce myself, Suleima Tang Martinez. I'm retired from the University of Missouri in St. Louis. I guess my question is, am I correct that the frogs are mating with more than one partner at a time or not?
Yeah, they seem to be pretty promiscuous. Yeah.
Okay, great. So then the question I have is whether there's any evidence like these frogs are depositing the younger, smaller tadpoles sort of preferentially in pools that have large tadpoles. And given that they seem to have this pretty amazing cognitive skills and so on, I wonder if one of the things that the fathers are doing is keeping track or remembering whether this particular pool where they decide to deposit the smaller tadpole is occupied by one of their other offspring, maybe full sibs of the one that it is depositing. Because if that's the case, and the full siblings are much less likely to cannibalize each other, then they could say, well, yeah, I'm going to put it here with this big one. My previous offspring has done super well, and this is a full sib, so I'll deposit it there, sort of on the assumption that there will be less likely to be cannibalism.
Absolutely. We had the exact same thought. And so actually what we did is that we took the tail clips of all the tadpoles within a pool, and also a toe clip of a dad. And so we're actually running those relatedness analyses right now to see what's the average relatedness within pools. The one issue that we are running into is it requires quite a lot of genetic information. And so to get enough, we assume that the adult transporting a tadpole is their father. And so we have used that as a genetic control, but you need to have like quite a few of those. And so of course we're limited with the numbers that we're allowed to clip and catch. And so, but what it's looking like right now, and I don't want to, speak is off the record, but it seems that the relatedness is not as high as we thought it would be.
Interesting.
Yeah. And so either that's because we don't have enough data and we need to rerun the analyses and we need to look at this from a different perspective, or maybe there's something there. Also, the genome of poison frogs is huge. It's so big. And so it requires the markers. I mean, I'm not well versed in genetics at all, but apparently the markers and the genetics, there's a lot going on there that needs to be, they need a lot of information. So yeah, we'll see.
Thanks. That answers the question. I mean, I think it's a really interesting possibility. The other question I have is whether anybody, I guess from your talk, I was really surprised by the ability that these frogs are obviously able to form maps on sort of 3D maps and remember, where their tadpoles are and all of this other stuff, which is pretty sophisticated cognition. And I'm wondering if there's anybody that you know of that's actually studying cognition in these frogs, because it sounds pretty amazing.
Yeah, Lauren O'Connell, who is based at Stanford, is doing a lot of really cool brain stuff right now. They also are working with Ranitomea, which is another kind of poison frog that has the Oophagus poison frog, and so mothers will deposit unfertilized eggs into bromeliads, and so they're also looking at the brains of the moms and the tadpoles, and there's this whole begging ritual that goes on. So I'm sure they're going to come up with some really cool stuff about that.
Yeah, and what's the name again? Lauren
O'Connell. It's
O'Connell.
Yeah, CONNELL, I believe. But they're stars out there. They're really cool. Actually, at that point, one thing that kind of gets tied up in that is the question of how do dads find these arboreal pools?
Yeah.
Do they just like randomly climb up these 20-meter trees looking for a hole at the top? How does this happen? And so we've throughout time, there's been many trials or attempts to think about how these dads are finding these pools. The most substantiated, untested claim is the thought that Trachycephalus, these big milk frogs that I was showing before that breed up there, they also call from those pools. And when that's actually how we find them in the jungle. If you put your hand on the tree where they're calling from, you can feel the vibration moving through your hand. And so again, completely unsubstantiated, because some people have tried to put like giant speakers at the tops of trees playing Trachycephalus calls and nothing has happened. So I don't know if it needs to be multimodal or something else, but it is quite fascinating, not only that these frogs are able to orient and navigate space, but also how they find pools is just, it's so mind-blowing to me that it's really, we're not quite sure.
Yeah. Thank you. So really a fascinating talk and a fascinating system.
Thank you, Suleima. Our next question comes from Eduardo.
Thank you. I'm Eduardo Fernandez Duque from Yale University, currently joining you from Argentina, where I'm on leave. I think I want to second some of the previous comments. Thanks for a very, very fascinating introduction to this system. Your enthusiasm is contagious. So thank you so much for making listening to you so much fun. And I'm delighted. We need so much to cross boundaries between big groups of animals. I'm a mammologist primarily, even more primatologist. So, and I do work on parental care, and these opportunities of kind of opening my mind to how we think about parental care in other talks are just so needed. So thank you for that. Along those lines, I want to follow up on Carson's question, but before I do that, if I remember well, you had a slide early on saying that in mammals, 100% of parental care is done by females. I don't know if I got it wrong, but that is not correct in any way.
That was it was 100% of mammals provide care. I misread that. Sorry.
It must be it is probably my father angle jumping on that, because males and males, human and non-human, are doing an awful lot for parental care, which takes me to follow up on Carsten's question about the lazy father. How well can you follow tadpoles? Because those males who are, I don't want to attribute intentionality to what they're doing, but what you know, and you have shared so nicely, some transfer all 3 tadpoles in one trip, others transfer. But what we really need to know is how those tadpoles fare. So it's not enough to look at what the dads do. So my question is, how well suited is the system to really look at what matters, which is tadpole survival? Exactly. That's what we need to examine in detail. It's made me think about the talk we just listened to on baboons, male care. Unless we know how the infants do, tadpoles are infants, it's going to be really hard to identify the adaptive value of the dad's behavior.
Absolutely. I started out my PhD with big dreams. And so I actually developed a method to be able to track tadpoles. So by injecting these visible elastomer tags that are fluorescent and UV, I was able to tag tadpoles that had hatched before they were transported. And I thought, this is great. I'm going to be able to watch where the dads transport them, because I'll just shine a UV light into a pool and easy. And so then I brought all of the stuff to the field, and it turns out that finding clutches of eggs is actually really hard. And not only that, but once you find a clutch of eggs, then you have to see where the dad transports them. And sometimes he transports them to the tops of trees. And I'm 1.64 meters, and I was like, well, this is not working. So I think within a wild system, my original dream is not going to happen. However, I do believe that we can use this tagging system in a laboratory setting, perhaps using larger terraria that allow for some more diverse kind of deposition sites, and see from there. And that's really where I think hopefully we'd be able to start untangling those kinds of questions. Also, sorry for misspeaking at the beginning. I don't mean to shame male mammal parents. I know they're there fighting the good fight.
Yeah, and I just, this is not a question, but if we run out of questions, I think that Tomislav question, it's something that I don't think has come up enough during the fine meetings and the questions and answers, but having an internationally informed discussion, debate about the ethics of animal research without, and I think we'll need to force ourselves not to approach it as scientists, but human beings, because it's mind-blowing. But I mean, I agree with Tomislav. It's fascinating. We may find ourselves having very intense discussions, and that is fine. I think it would be fantastic to hear what each of us are learning from our own countries and cultures, and how much we end up doing what a few countries are telling us to do when this is not a scientific decision exclusively. So if there's time, I'd love to hear from other stories. I have my own about what Argentina wanted me to do and the US want me to do, et cetera, et cetera. Great. Thank you so much.
Just on that point, though, I mean, at least for us, cannibalism is a natural behavior, right? Like if you just walk around in nature, it's happening, and it happens when tadpoles are fully fed and have plenty of space. So it's not like they need to be starved for this behavior to happen. It's not like they are, you know, placed in tiny little cages and we're forcing it to happen. So that's all I'll say about that now until we want to open up that can of worms. But also, like you said, between countries, between Finland and then even Austria, completely different expectations, completely different definitions of what pain is. But again, very wormy, box of wormy conversation that we could talk about for many, many hours, I'm certain.
Do that. Thank you, Dardo. Our next question comes from my behavioral ecology class.
Hi. My name is Sofia Garrido. I'm from Universidad del Rosario here in Colombia. I'm an undergrad student, actually. Adriana is a student. And I didn't have the chance to be throughout the entire talk. However, I've read some of the articles that were suggested, and I have one particular question. In those articles, they mentioned that there are some species of poison frog that seem to have tendencies to deposit tadpoles in pools with potential cannibalism. However, do you think that there are like two possible behaviors in these frogs when the tree gaps occur? Like, for example, one where some of them tend to be pioneers and fast to occupy those spaces, or do you think that like individuals way to evaluate the resource quality in these novel sites? Like, what's your perspective on that behavior and like how that happens?
That's a brilliant question. Thank you so much for asking it. So I think that when a tree fall happens, an amount of resource, so when this tree falls, right, all of these tree holes suddenly are available for the pickings. And I think because quality resources in the system are relatively sparse, that we do just have this influx of dads that arrive depositing their tadpoles. So I don't think that there's some that prefer tree gaps or not. I think because they're so unpredictable, they just kind of happen and then everyone just kind of jumps on it. But then I do think there's probably some luck involved. You know, whoever's closest to the tree fall is going to be the pioneer, right? Like if you know you're 10 meters away, then you're going to get there first. So I think that's definitely probably, at least from my perspective, maybe what's driving some dads arriving there earlier than others. Did that answer your full question? I'm sorry if I missed something. I got a little excited.
Yeah, that was perfect. Thank you very much.
Thank you, Sophia. We have another question from Tomislav.
Thank you, Eduardo. I just wanted to connect to your finale because as a coda to this wonderful talk, which I thank for, I think it would be very interesting if you or we could make a panel or forum, discussion forum about what does it mean, the conception of animal welfare. I mean, we are talking here about a scientific project which is stopped. I mean, scientific project which aims at investigating cannibalism in poison frogs, and which is stopped if frogs are cannibalizing each other.
Yeah.
I mean, we are here. I mean, it is my submission that something interesting at the traits. I mean, okay, I understand, but in the long run, it comes to the question. I mean, what is the consciousness? I mean, who has the consciousness? And I hope you understand what I'm aiming at. But I mean, if you are investigating cannibalism in a poison frogs, and you are hindered them to cannibalize each other. I mean, shouldn't we talk about it on the greater global scale? That was my question. That's all.
I agree. I think that's why I think it can be. I think it's the kind of topics that we should not avoid. I'll say just a few more words because I'm seeing Suleima in the audience, and Suleima has been active on this, has written about it, has thought about this much longer than I have. I think that there's a benefit as human beings, as scholars, as intellectuals, to engage in such a dialogue. I don't think that, personally, I would not expect that we will reach any conclusions. I don't think that conclusions would be valid based only on evidence-based data, because it's what you're saying. For me, it's more, let's play amateur philosophy or amateur ethics. It's what is pain? What is welfare? What is but we do need to make some decisions, us or our institutions.
Yeah.
No, yes, I think I'd love to hear from others. I would definitely invite everyone to understand that one should walk into this kind of meeting, if we had one, doing what we preach, right? I mean, if we're going to be having these conversations, and you belong on the side where you're preaching about diversity all the time, you have to be ready to welcome diversity of opinion about pain, welfare, and ethics. That's diversity. Diversity is not just as long as it fits my ideas, but when we start talking ethics in different countries, in different cultures, I mean, we could not execute a National Geographic grant because we couldn't get the US-based ethics committee to approve of our protocols for handling the chicken that we were going to use as live bait. Right? And this is in a ranch where we were giving the chicken to the people to eat after the permit, right? So who is going to decide what happens with the chicken in a ranch in Argentina? Is it going to be a bunch of scholars in an Ivy League institution in the US? Do we want that? It's I've been part of just 2 examples. We had a major problem in the primatology journal because you had people in Brazil collecting a specimen of a non-human primate with permits from the country, approved by every single level of the country, and now a journal based in the US says, we don't want you to do that. So, Suleima, you must have an awful lot to contribute to any such debate.
I think I probably do, and I should, just so that we're all on the same page, I should say that I was one of 2 people who drafted the original guidelines on animal care for the Animal Behavior Society, way back in the Precambrian. But I have had huge issues. And I'll give you just a few examples, and these are the things that have really bugged me and where I think that guidelines have been misused. At Berkeley, my former PhD advisor was doing research on wood rats, and wood rats go out and collect leaves and branches and grass and all kinds of things, and they build this huge dens, just made up of stuff that they've collected from the environment. And this is Steve Glickman, for those of you who might know of him, but Steve had kept a colony of wood rats in the field station, and the animal care people essentially required the area that the animals were in, which was quite large, to be cleaned out once a week, so that once a week all of their homes were destroyed, and that you were supposed to sterilize anything that went into the space, and essentially ultimately it killed the study, because you can't do that sort of thing. I myself had running battles with our animal care committee at University of Missouri, St. Louis, because I was working with prairie voles, and prairie voles are very social. They are highly dependent on odors. And again, they insisted that in terms of the lab colony, that they be kept in the same sorts of conditions that they keep, mice and rats for medical research. And this was incredibly disruptive to the animals. Their cages were supposed to be cleaned twice a week, which meant everything was taken out, everything was, deodorized, everything was sterilized. I mean, it was insane. And they also had limits. You could only have like, I can't remember now. I think it was 2 animals or 3 animals, not counting offspring, together in a cage. And what was ironic is that at one point I did this big experiment using this big experimental behavior room. And I was putting in groups of voles, either related or unrelated, into this room. And each vole, I had put in enough nest boxes so that each vole, it was 6 voles total in each in each one of the trials. And I had put in 6 nest boxes so that they could all have their own nest box and so on. And one day I came in and my voles were gone. I couldn't find my voles anywhere. And I was like, oh my God, did they escape? What happened? I looked all over, and I happened to look down at the base of the door, and the base of the door was built in such a way that there was a tiny little area along the bottom of the door, like maybe 2 inches, like a little square metal, but it was hollow. And I see these little tails hanging out. All the voles had crammed themselves into this tiny little space when they never, among the related individuals, typically I found them jammed into one of the nest boxes. All the other nest boxes were not being used. And it was crazy that we had all of this regulations about in the actual colony about how to keep the animals, what to do with them, and so on. And I actually felt, and what I argued is that you can't have guidelines for wild animals that have a very different life than the sort of artificial, mice that you have in medical experiments. You can't have the same conditions for them. But it was a struggle like that went on and on endlessly, and I would bring in this evidence that no, my animals have to be together.
I think this is the crucial question. I think this is a crucial question that is raised now, because I mean, animal behavior and animal welfare are in the core of our science, besides evolution. And that what you mentioned now is just an example of development of ethical thinking about animal behavior that is really worthy of discussing. So that is my suggestion to Eduardo and to Karsten and to Adriana and to all of you who are organizing this. It is quite interesting, I think.
To this.
That's a great idea, Tomislav. I think we're going to consider that within our non-seminar, and perhaps like a kind of symposium might be a good idea. So thank you, Tomislav, for your idea. We have another question. We have 3 more questions in the chat. So next one comes from my class. So behavioral ecology. There you go.
Hi, my name is Camilo Arenas. I'm also from La Universidad del Rosario. And I have a question related to one of the suggested articles. And it said that sometimes in one pool there are multiple species or multiple tadpoles of different species. So I had a question, and it is, if in pools where there are tadpoles of multiple species, is it more common to see cannibalism or predation by these other species?
Yeah, great question. I actually haven't measured this in the lab for obvious reasons, but I would say my gut instinct when looking at these natural systems is that they opt for predation first, and then cannibalism would occur. Because also, I mean, the tinctorius tadpoles, they're quite fast. They're good ambush predators, so they're pretty good at hunting others. So I would also substantiate that they definitely go for predation first. I wanted to maybe build onto what others have been commenting about this animal welfare thing. Just to make a small aside, that there's also there's like also a veterinarian approach versus a field biologist approach, which I think you guys have been kind of echoing here. And I think that also kind of adds a layer of, if we think about field biologists and maybe field knowledge, which then we could even, think about even indigenous knowledge and how these people like see the world and see animals and interact with the natural world. And then also the veterinarian approach. I think there's a lot of communication and cultural clash there, and a lot of information that's maybe not being valued or being seen at an equal level. I know a lot of field biologists from Colombia who have worked with frogs for many, many decades. And then when put up to a veterinarian from an official institute, apparently their knowledge means nothing. And I think that that's worth commenting on and dissecting as well, and how we value knowledge and how we value the people that say the knowledge. I think that's really worth examining, because I think there's some other layers to unfold there.
Thank you for your answer.
Thank you, Camilo. We have another question from Kaya.
My question became irrelevant as the discussion went on, but I really like that last point, Chloe. I think that's central to actually this whole bioethical question. And I think it would be really interesting to have a broader discussion with some dedicated time, especially on the cross-cultural approaches, and also how they clash with the sort of Western science, whatever, industrial complex, because there's like so much pressure to produce and to, publish and to just get it done, and not necessarily enough emphasis and concern for not only the animals' well-being, but also the people's well-being that are doing it. Like I know a lot of people that went into biomedicine and like just were like forced to do horrible things to mice and like never got over it, And so there's that whole side of it.
Yeah, absolutely. I took an animal welfare class in Finland. They taught me like 50 different ways how to kill a rat. And it was so sad because we spent the entire morning learning how to handle rats. And then like one fell asleep in my lab coat and they're like, all right, euthanasia. And it's like, wait, what? Of this guy in my pocket? It was intense.
Yeah, that's quite true. In Germany, you have to go through one of those courses to be able to handle your animal. And I didn't have to take it, but from my friends, they said that you have to learn how to kill them in order to be able to take care of them. And I was like, okay, wrong approach, but yeah, that's sort of the law. You need to take that course if you want to do animal experiments. Suleima, you have a question?
No, I just wanted, since we're on this topic, I just wanted to alert people to the fact that many, many years ago, and I don't actually have the reference, but there was a paper that was published by a Japanese scientist who was doing, spending a sabbatical in the United States. And the paper was titled Sushi Science and Hamburger Science. And it dealt very much with how culture can affect how you approach science and what you think science is, and the role of animals and all other things. But I love the title, hamburger science or sushi science or hamburger science, or the other way around. I can't remember exactly, but I think it speaks very much to this, to this issue of cultural differences and how they affect science. And of course, I'm sure Eduardo is probably very, very familiar with this. The Kyoto School of primatology and how their approach differs so much from the typical Western. You go out with your hypothesis and then you test your hypothesis versus, collecting the data, observing the animals, and then seeing what comes from that, which of course can then generate hypotheses. But again, I think cultural perspectives are fascinating in not just in animal behavior, but even more broadly. I just wanted to mention that.
Agreed.
Thank you, Suleima. Okay, I don't see any other question, and we don't have questions in the YouTube channel, so I'm just going to